Hello students, in the last lecture we have studied fractional distillation using the fractionating column, how two liquids which differs in their boiling point are separated, that yesterday or in the last lecture we have studied. So fractional distillation is a process which is used to separate the two liquids which differs in their boiling point by a small difference. Then at that time we have to use a fractionating column. We have to use a fractionating column if two liquid differs in their boiling point by less than 30 degrees Celsius. That yesterday we have studied, and principle of that separation, that is the more volatile liquid distilled out first, leaving behind the less volatile liquid. Today we have to study some more process of extraction. That is, first of all we have to study solvent extraction, and later we will study what that is the chromatographic technique. In chromatography there are two types of chromatography: adsorption chromatography and partition chromatography. So here see solvent extraction. In this solvent extraction, when an organic substance is present, when an organic substance, organic compound is present in aqueous solution, when organic substance present in aqueous solution, then for the separation of that organic substance from that solution, the technique used is solvent extraction. Technique used is solvent extraction. Here see, in this first, what that is the funnel, I have taken organic compound in aqueous layer, and this is the solvent layer. Means first of all we have to take that liquid or that substance which is with aqueous solution or which is in aqueous solution. When that substance is taken, then what to do, that is the, in that organic compound in aqueous layer you have to add the suitable solvent. Suitable solvent, that is the add organic solvent, suitable organic solvent in that aqueous, what that is the layer containing the organic compound, in which that organic solvent, that substance is more dissolved, that substance is more soluble. So such solvent we have to add in this aqueous layer containing the organic substance. Okay. When that organic substance is vigorously shaken with the organic solvent, then the organic substance is now miscible in that organic solvent from the water. म्हणजे तो सबस्टन्स असा त्याच्याबरोबर ही शेक करायचा आहे, that is shaken with the organic solvent in this funnel. In this, what that is the solvent extraction process. Here first of all we have taken organic compound in aqueous layer, then organic solvent is added in it, and it is vigorously shaken with that organic solvent like this. Then what happens, this substance from organic substance from the aqueous layer is miscible in organic solvent. Okay, when it is miscible in organic solvent, then here you see organic compound in solvent layer. अभी इसमें क्या हुआ कि तो सबस्टन्स त्या aqueous layer मधून organic substance मध्ये आला, organic solvent मध्ये आला, and aqueous layer is separated. Aqueous layer is separated. Now here we can separate this aqueous layer by using this stopcock. When we start this cock, then we can collect this aqueous layer in one beaker. And what is left here? Organic compounding solvent layer is left. Then organic compound from organic solvent can be separated by distillation process. By distillation solvent is removed. And you will get what that is the solute is obtained or formed. So that is nothing but the solvent extraction process. Once again I am telling to you, when organic substance present in aqueous layer or it is present in water, then by this process we can separate the organic substance from that aqueous layer. How? That is, first of all we have to add a organic solvent into that aqueous layer containing organic substance. Then it is vigorously shaken with that organic solvent. Then what happens? The substance from that organic compound, substance from that aqueous layer is dissolved in this organic solvent, and again there is formation of two separate layers. That is the aqueous layer and this is the organic compound in solvent layer. This aqueous layer can be separated or it is removed by using this stopcock. What is left? That is the organic compound in solvent layer, it is left, and solvent from this organic compound can be removed by using the distillation process. Repeated distillation takes place and that solute can be separated from that, means solvent is evaporated or distilled out and solute will be left. In this way that substance is extracted or obtained by this solvent extraction process. Now see the next one technique for the separation of component, that is the chromatographic technique. This chromatographic technique is discovered by the Swiss chemist. Chroma means color. Here chromatography means what? That is the chroma. Chroma means color. So it is based on the formation of color bands, color spots in this process are formed, and therefore this technique is called as the chromatographic technique. This chromatographic technique consists of two phases. One phase is mobile phase and one is stationary phase. Stationary phase is act as adsorbent and mobile phase contains the mixture of component, and that mixture of component is separated by using this adsorption chromatography technique. So there are two types of chromatography, that is the adsorption chromatography, and second technique later we have to study, it is partition chromatography. Now in adsorption chromatography there are two phases, that is one of the phase is mobile phase and another phase is stationary phase. Stationary phase is either a paper or a chromatographic plate, or it is also silica gel, alumina gel. Such type of the adsorbent are used as the stationary phase. Here you see, first of all we have to study column chromatography. In this column chromatography, first of all we have to construct a column. How column is constructed here, you see. This is a long elongated glass tube, right? At the bottom of this long elongated glass tube, there is what that is the glass wool plug. Glass wool plug is what that is the plug at this bottom. Then this this long elongated glass tube is filled with silica gel or alumina gel slurry. Okay, and in this way it is completely filled with silica gel or alumina gel. Silica gel or alumina gel slurry, it is filled in it, and in this way what is that is, this is nothing but the column stationary phase is constructed. It is manufactured by filling silica gel or alumina gel in it. This is the stationary phase. Stationary phase. Stationary phase. Stationary phase, it is the adsorbent. Now, suppose you consider mixture of the component, that is the A plus B plus C. We have to separate by this technique. One point here you have to remember, when components are present to very small amount or the quantity, very small extent, then at that time for their separation chromatographic technique is used. Chromatographic technique is used. So mixture of component A, B and C, we have to separate by this column chromatography. So first of all we have prepared a column. How? That is stationary phase means silica gel or alumina gel slurry is filled in it. Now our column is prepared. Now this mixture in suitable solvent, then that mixture of A plus B plus C plus solvent is poured at the top of this column. म्हणजे ते मिक्सचर इथे आपण ओतायचे. When we pour that mixture, then that component going down, while going down they get adsorbed on the stationary phase or adsorbent. But what happens here, you consider, suppose rate of adsorption of A is greater than B and B is greater than C, suppose. Then what happens, while going down this mixture along with the solvent, the rate of adsorption of A is greater than B. So suppose you consider from this point A starts to adsorb, and totally in this, what that is the portion, the component A get adsorbed. Here component A get adsorbed. Now B plus C going down. काय झालं, एवढ्या पोर्शन मध्ये कंपोनंट ए ऍब्सॉर्ब झाला आणि बी आणि सी खाली चाललेला आहे, कारण त्यांचा रेट ऑफ ऍब्सॉर्प्शन हा ए पेक्षा कमी आहे. नाउ अगेन व्हाइल गोइंग डाउन, आफ्टर सम टाइम व्हाट हैपेंस, द रेट ऑफ ऍब्सॉर्प्शन ऑफ बी स्टार्ट्स, एंड हियर within this portion what happens, that is the total B get adsorbed. समजा या पोर्शन मध्ये सगळा B काय झाला, adsorb झाला. Here it forms, A forms separate column, specific color. Now B form the separate column and specific color. Now what is left? C. Now C is going down the column, and while going down the, what that is this, then C starts, suppose from this point to up to this adsorption start, and within this portion the C totally get adsorbed. And you see here there was 3 component in the liquid in the mixture, and that 3 component present in the mixture are separated by this column chromatography. How? That is, this mixture in a suitable solvent is poured here. Rate of adsorption of these 3 components are different. First of all, rate of adsorption of A is greater than B and C. Therefore, A starts to adsorb from this point, and within this portion it adsorbs. Here there is formation of a specific color band, that is the A forms. While going down, the B adsorbs here, it forms a specific color band, and C adsorbs here, it also forms the specific color band, and hence we can identify from the color what type of ions, radicals are present in that mixture, or what are the components present in that mixture. So this technique is called as chromatography, because here there is formation of specific color bands. Color bands are formed, and therefore that is called as the chromatography. Chroma, color, graph, column. Therefore it is called as the chromatography technique. So column chromatography. Now you can separate this. If we heat this column, the reverse process of adsorption takes place on heating, because this is physical adsorption. You remember, in adsorption and colloids we have studied, the physical adsorption is inversely proportional to temperature. So if we increase the temperature of this column, then desorption starts. The process of removal of adsorbed molecule from the surface of this adsorbent is called as the desorption. So desorption starts. Now whose rate of adsorption is less, its rate of desorption is high, and therefore in first one flask or beaker C collected, later B get desorbed in second flask or beaker B collected, and in third flask or beaker what is collected, that is the A collected by using this stopcock. And in this way the mixture of component present in this mixture, that is A, B and C, first of all identified from their color, and then they are separated by using this chromatographic technique, that is nothing but the column chromatography. It is column chromatography. Now see, second one, chromatography, we have to study thin layer chromatography. In this thin layer chromatography, first of all we have to prepare thin layer chromatographic plate. Okay, TLC plate. So what is TLC plate? It is very simple. This is a glass plate, right? This is a glass plate. On the surface of this glass plate, adsorbent in this way spread. In this way what is spread? That is the adsorbent spread. This is nothing but the TLC plate, and from the bottom or from any side at 2 centimeter distance, one spot of that mixture of liquid component is given like this. काय करायचं आहे? We have to take a plate. On that plate adsorbent is spread. When adsorbent is spread on that plate, then we have to take a mixture of that component. That mixture of one spot of the mixture of component is given as a dot on that TLC plate, and now this TLC plate is poured, means placed in a beaker containing solvent, right? TLC plate is placed in a beaker containing solvent, and when it is placed in a beaker containing solvent, here see what is the level of solvent, that is called as the baseline. This is nothing but the sample dot, spot or the sample dot, and what happens, this is the adsorbent coated on a glass plate, this one adsorbent coated on a glass plate. Then what happens when we place this plate in the solvent, then this solvent starts to rises up. What happens? Solvent starts to rises up. When this solvent starts to rises up, then when it comes at the spot, sample spot or the dot, then it rises up along with that dot. ते त्या डॉट मधील कंपोनंटला घेऊन राईज होईल आणि पुढे गेल्यानंतर जसा त्यांचा रेट ऑफ ऍब्सॉर्प्शन असेल, हियर इन दिस वे देयर इज फॉर्मेशन ऑफ द स्पॉट्स ऑन that TLC plate on the basis of adsorption. Suppose rate of adsorption of this sample or component is more, it get adsorbed here, second get adsorbed here, and third get adsorbed here, and solvent rises up. Solvent rises up, and finally this is the highest level of solvent that rises here. Okay. Now this TLC plate, on this TLC plate there is formation of very small colored dots, spots, and by using that colored spot we can identify what are the mixture or what are the component present in this mixture. आपण त्याला फाइंड आउट करू शकतो त्या डॉटच्या कलर वरून की कोणता कंपोनंट आहे. इफ दॅट स्पॉट फॉर्म हॅव नो कलर, then you have to place this TLC plate under ultraviolet light, then that spot will show the fluorescence. It चमकतील fluorescence, and by using that we can identify the component present in that mixture, and this technique is called as the thin layer chromatographic technique. From this thin layer chromatographic technique we can identify what that is the, we can determine retention factor. Okay, retention factor, how will you determine that we have to study. Here retention factor means here spot obtained, retention factor of that substance. This is nothing but the line, distance traveled by from the baseline of solvent, आणि समजा हे स्पॉटचं डिस्टन्स आहे, this is x, and distance traveled by the solvent is y, and retardation factor, see the formula, retardation factor R is equal to distance traveled by the solute from the baseline, distance traveled by solute from baseline divided by distance traveled by the solvent from baseline divided by distance traveled by solvent from baseline. So that is nothing but the retardation factor of the liquid component, or that component can be calculated. Migration of solute, here there are 3 solutes suppose in this case, then this is nothing but the one solute, distance traveled by this solute divided by distance traveled by solvent from baseline, distance traveled by this solute divided by distance traveled by solvent from this baseline. So that factor is called as the retardation factor. That is known as the retention factor. Sorry, it is nothing but the retention factor. It is the retention factor, means how much is the ratio of distance traveled by the solute from baseline divided by distance traveled by the solvent from baseline. So this technique is called as the thin layer chromatographic technique and column chromatographic technique. Last one point remaining, that partition chromatography, and some more points we will continue in the next lecture.